The CRISPR era is not just upon us, it's rewriting the book of medical science. A revolution is taking place in laboratories across the globe, and it's called CRISPR. Clustered, regularly interspaced, short, palindromic repeats. This mouthful of a term represents a simple yet powerful tool for editing genes. CRISPR, in essence, is like a pair of molecular scissors, capable of cutting strands of DNA with unprecedented precision. It's a game changer, allowing scientists to alter, delete, and replace genes in any organism, including humans. Picture this. It's like editing a manuscript, but the manuscript is the genetic code of life. This revolutionary technology holds immense potential. Scientists believe that with CRISPR, we could cure genetic diseases, create hardier plants, and even eliminate pests. Imagine a future where conditions like cystic fibrosis or sickle cell disease are no longer life sentences but curable with a single treatment. CRISPR has opened a new chapter in our understanding and manipulation of life's blueprint, the DNA. The story of CRISPR is one of science, perseverance, and groundbreaking discovery. The tale begins in the late 80s, when scientists noticed something peculiar in the DNA of bacteria, strange repeating sequences interspersed with unique spaces. They named these mysterious patterns clustered, regularly interspaced short palindromic repeats, or CRISPR for short. But what were they for? It took years of relentless research to uncover the truth. These were not just random sequences, but a part of the bacteria's immune system, a genetic memory of past viral invaders. Fast forward to the early 2000s, when researchers discovered associated proteins, aptly called CRISPR-associated proteins, or CAs. The most famous of these, Cas9, would soon revolutionize gene editing. It was the molecular scissors that could cut DNA at precise locations, guided by the sequences stored in CRISPR. This was a remarkable discovery, but the true breakthrough came in 2012, when two scientists, Jennifer Doudna and Emmanuel Charpentier, adapted this bacterial defense mechanism into a versatile gene editing tool. They realized that by designing their own guide RNA, they could direct Cas9 to cut any DNA sequence they wanted. The implications were immense, and the scientific community was quick to seize upon this new tool. Within just a few years, researchers were using CRISPR to edit genes in everything from bacteria to plants to human cells. Diseases that were once thought untreatable suddenly seemed within our grasp. In 2019, the first clinical trials using CRISPR began. These initial trials aimed to treat genetic diseases like sickle cell anemia and inherited blindness. While it's too early to declare victory, the results so far have been promising. In less than a decade, CRISPR has transitioned from a microbial defense mechanism to a cutting-edge tool in biomedicine. We are living in the CRISPR era, a time of unprecedented possibilities in medical science. We've journeyed together through the revolutionary advent of CRISPR and its transformative journey. Now, let's dwell on the future that beckons us. This era ushers in a world where genetic disorders could become a thing of the past. Diseases that have plagued humanity for generations could be edited out of existence. But it doesn't stop at disease eradication. Imagine a future where we could enhance our natural abilities, stronger bones, sharper eyesight, or even heightened intelligence. But with great power comes great responsibility. The ethical implications of CRISPR are just as vast as its potential. What are the moral boundaries of genetic editing? How do we ensure its use for the benefit of all, and not just a privileged few? And what about unintended consequences? These are challenges we must grapple with as we navigate the CRISPR era. As we stand on the brink of this new era, the question isn't just what CRISPR can do, but what we will choose to do with CRISPR.